Okay, good.
Everyone, recording.
Hi, everyone. My name is Josephine, and this is Annalyn. We're going to talk about scorpions and their chemistry. So we wanted to first start off about chemistry of scorpion venom. So venom does consist of certain chemicals and amino acids, such as their venom being toxic and harmful to one's health, causing it to lead to poisoning in the bloodstream, aspiration, shortness of breath, cardiovascular diseases, and mortality. So there are 66 amino acids that are present, 20 of which we learn in biology, such as valine, leucine, and isoleucine. The molecular weight is about 3,400 grams, and the chemical structure consists of 141 carbons, 238 hydrogens, 46 nitrogens, 39 oxygens, and 6 sulfurs.
And this is a short background of scorpions. They have been around for over 400 million years, live and adapt a majority of their species from desert areas. Thousands of species, some which of are more deadly than others, and today the living families are very low, roughly 22 species, and could be more. A gallon of scorpion venom consists of gamma hydroxy butyric acid for cost like 2,500, and this is a rare antivenom, and they range from 2 to 6 years of life, and they isolate themselves from the smells of peppermint, lavender, and cinnamon. Why this topic is important? It's because from those 22 species of scorpions around today in the world, some of them can cure certain and specific causes of cancer, like brain tumor, breast cancer, and arthritis. The venom categorizes in apoptogenic and promotes death at specific phases in the cell cycle. Cytotoxic kills the cells that cause cancer and stops cancer cells from dividing or multiplying. In other cases, it can shrink in size. And immunosuppressive reduces the immune system to respond to intruders like foreign cells.
So as for cancer cells and venom, there are some bioactive molecules that help target those mutated cells and break them down. There's 4 potential mechanisms are induction of cell cycle arrest, growth inhibition, and apoptosis. The second is inhibition of angiogenesis. Third is inhibition of invasion and metastasis. And the fourth one is blocking specific transmembrane channels. There are, in this image, you can see that it shows the bioactive peptides in scorpion venom that does help. So it modulates and blocks the ion channels, induces the apoptosis and cell arrest in the cancer cells, repolarizes inflammation-induced cancer properties, inhibits angiogenesis, and inhibits invasion and metastasis of the cancer cell. In this next one, it shows the purpose of cancer cells and venom. Basically, it's showing the transformed cell inherits mutations and acquired mutations involved, such as chemical agents, viral infections, and radiation. The cons are such that it does cause chronic inflammation and oxidative stress. So in this image, you can see that the first one does show unbrittled cell proliferation, which is basically showing the activation of telomerase and alters the ion channel. The second one is the evasion of apoptosis, which breaks down the p53 genes and the caspases genes. And the third one talks about being sustained angiogenesis, which is overexpressed specific genes. And the last one is the tissue invasion and metastasis, such as altering the ion channels again. In this image, it's very short and simple. It does show that it's the breakdown of the venom being extracted from the scorpion, and it then is reinserted intravenously. And another example was brain tumors. We did want to show a video, so I hope it works. I'm not sure if the sound will work, but we could just see the pictures. And I'll debrief on this video in just a second once it's over. So they're doing biomarkers basically of venom and cancer cells. And now they're doing the biomarkers with blood, and they will mix it in with the venom as well. So the CAR T cells do come in contact with the cancer cells, and it does destroy it, or in hopes of destroying it.
This was the first time that anybody proposed doing this type of thing, using a toxin for this purpose. And so it was an invention. And at every step of the way, we were waiting for something to go wrong. And it didn't. And so we're now at the point where we've done everything in terms of characterizing it and doing safety studies. And there's only one thing left to do, which is to use it in a clinical trial and trial in a patient.
We're really excited about what we're going to learn and how we're going to advance the field for the treatment of brain tumors.
Okay. So in that video, they basically went on the City of Hope, what caused was trying to structure a sting operation. They're in hopes of having CAR T cell therapies for cancers, and they want to treat about 500 patients. And they were trying to test this since the 1990s to have CAR T cells and cell therapies be acting as a helping agent. So the first administer of the CAR T cells was to inject it in the tumor site and intravenously inject patients. And it did involve some chlorotoxic protein from the scorpion venom, which does target those chloride receptors for tumor cells. And their phrase that they kept going by was scorpion to immunotherapy. And so basically in this image right here, it shows how they remove the blood from the patient to get the T cells. From those T cells, they want to make the CAR T cells in the lab, so mutating it to make millions of them, and then infusing those CAR T cells back into the patient, which then those CAR T cells would bind to the cancer cells and kill them.
And next is the breast cancer, and it's the second leading cause of cancer-related deaths among women in the United States. Component, just like Josephine said, the chlorotoxin is used to facilitate specific delivery of the therapeutic agents directly into breast cancer cells. And chlorotoxin is a scorpion venom-derived peptide. It may facilitate the entry of chemotherapeutic compounds into tumor cells to target drug delivery and improve efficacy. Preliminary assessments indicate that the scorpion venom are inducers of apoptosis in breast cancer cell lines. It recognizes breast cancer cells and enters these cells pairing with the therapeutic payload, which is a matrix composed of polymeric materials with multiple functional groups, which signal emitters. And then next is the arthritis. So researchers found that the drugs from scorpion venom that concentrate in joints may replace steroid therapies for arthritis. Venom compounds precise administration is enabling safer, more effective medicines that could change inflammatory disease management. And the mini protein found in the death scorpion can bind to cancer cells, but not the healthy ones. And the tiny protein rapidly accumulates in joint cartilage. These proteins with steroids reverse inflammation in rats with arthritis.
Okay, so to conclude, basically what this whole PowerPoint was talking about was relying on scorpion venom and other aggressive venoms to help cure cancer cells, arthritis, and other factors that do come into contact. So the proliferative characteristics by inducing apoptosis, it's not very cost effective because it is $2,500 for a gallon. And there are no trials fully completed. There are clinical trials still happening with ongoing studies. The venom does need to possess those target cells and reduce the toxicity. And those cancer cells are then, terminated to be deadly. And it is a cause of worldwide mortality rates for, many majority, like Annalyn said, for breast cancer, it's the #1 or #2 leading death in women. And CAR T cell therapy should be something that should go into our field of interest.
And then just a few fun facts about scorpions. They were around long before dinosaurs, and they're not insects and could be as long as 9 inches. Venom can include dozens of different toxins and have 2 body regions, 4 pairs of jointed legs, and an open circulatory system.
And these are our resources. Does anyone have any questions, comments, or suggestions? Do we stop the recording? Yeah. Okay.
There's us recording.
Yeah, did you stop? Yeah. Stop.
Pause.
Oh, pause it.